Good evening. My name is Nicholas Hanle, and today I'm going to be talking to you about cell cycle regulation in the cell. This is a slide. I will post the link to this video once I have it completed. And the objectives for this lesson or this activity. First, I'd like to be able to explain the phases of the cell cycle. We got to understand some background stuff before we get into some detail. Next, we're going to describe why the cell cycle is regulated. We'll also describe the structure and function of the cycle-independent kinases. And then we'll look at the four methods in which CDK activity is regulated. We'll look at a couple examples of how CDKs affect critical proteins within the cell cycle. And then we'll review some research on how CDKs are used or affected in cancer treatments. So if we look at the cell cycle is the mechanism by which the cell grows, replicates its DNA, distributes those replicated DNA or daughter or chromosomes into daughter cells, and then goes through the cell division process. Overall, there's kind of like two parts to the cell cycle. We have interphase, and then we have mitosis. Between those two areas, there are four phases the cell will go through. We have the three interphase parts and the one mitosis. So if we look at G1 phase is the first part of interphase, 95% of the cell of the time in the cell cycle is spent in interphase, according to Cooper. Mitosis may be the most dramatic part, but interphase is where we're getting everything ready. So it's kind of like a vacation. You prep and pack and plan, and then the vacation seems to be the short part. So mitosis would be like vacation, and the interphase would be like prepping for the vacation. So if we look at this, interphase is made up of gap 1, the S phase or synthesis phase, and the gap 2 or G2 phases. The M phase of the cell cycle is the mitosis stage. Upon completion of mitosis, the cell would then go back into G1 and we start the cycle over again. In the G1 phase, the cell is metabolically active, and basically what it's going to do here is going to begin growing. We need to get the cell size. It needs to be bigger so that when we split, we have two cells that are roughly the size they need to be. The M phase, in the G1 phase. The S phase of the cell cycle, and again, this is still interphase, this is where DNA is actually replicated and prepared for that part of it. And then finally, in the G2 part of interphase, this is where growth gets completed, as well as a lot of the proteins necessary for mitosis are produced and gathered and ready to go. And then in mitosis, duplicated DNA is divided methodically into two daughter cells. So this is where you have, prophase, metaphase, anaphase, and telophase, as you can see in this diagram. And cytokinesis is part of this as well, where the two cells actually pinch off and separate, forming the two unique daughter cells. Not all cells actually go through the cell cycle. Some cells are in a dormant proliferation state, kind of goes off of the G1 phase. We call it the G0 phase. Basically, the cell is kind of dormant. It's metabolically active here. It's still processing things and all that, but it is not going through the cell cycle. And it'll stay in that G0 phase. Some cells stay there forever. Others will stay there until they receive a signal triggering them to divide again. So kind of if there's a cell injury, they might activate the cell cycle again to try to repair that. But if they enter back in from the G0 phase, they will come back into the G1 phase and pick up the cell cycle from there. Okay, regulation of the cell cycle. We want to have this because it leads to proper cell division. We want our cells to be effective and do their jobs the way they're supposed to. So throughout the cell cycle, there are little checkpoints and stuff where we can make sure that the size of the cell or the DNA is undamaged. We want to make sure all those things are proper and necessary so that we prevent daughter cells that are not what they need to be. A lot of times here, defective regulation is going to lead to cancers, uncontrolled cell growth. So the cell cycle gets stuck in the on button, and we keep making cells forming tumors, and those tumors can then have effects on the surrounding functioning tissue and lead to the results of cancer. Um, the cell's ability or potential to divide is essential for the survival of organisms. So we want to make sure that we're dividing and doing what we need to, and we do that through proper regulation of the cell cycle. Um, in this process, we got to make sure that certain biochemical processes and events are taking place for the right amount of time in the specific order they need to be in order to ensure the progression of the cell. And cell signaling is going to play a major role in coordinating these cellular movements through the cell cycle. So we're going to look more about that now. Okay, cyclin-dependent protein kinases, or CDKs. These are heterodimer compounds. They have two components, a regulatory subunit that is the cyclin part of it, and a catalytic subunit. Um, in humans, binding of the cyclin to the catalytic subunit it changes the conformation of the catalytic part, allowing the active site to be opened up conformation wise to receive everything it needs to. So it provides the active amino acid residues present to be able to be exposed to what they need to be exposed to in order to react properly. In humans, we have 10 different cyclin molecules, and we identify them with letters, so like cyclin A, cyclin B, and so forth. And then we have about 8 different types of CDKs, and we represent them with numbers, so CDK1, CDK2. Um, CDKs are going to regulate the cell cycle when they're activated by various cellular signals. The amount of a particular CDK or cyclin is going to fluctuate throughout the cell cycle. So different CDKs, different cyclin molecules are active at different points, and that's what this diagram is showing here. So in this case, cyclin E is most active between the G1 and S phase, whereas cyclin D kind of builds up all the way. It's most active during the S phase, and then it kind of goes back down. Cyclin A is most active in the beginning part of G2, and then cyclin B is most active in the beginning part of mitosis. So different cyclins play different roles at different points in time. So by regulating the CDKs, this regulates the cell cycle. There are 4 ways we can regulate our CDKs here. We can phosphorylate or dephosphorylate the CDK molecule itself. Secondly, we can control the degradation of cyclin subunits. Next, we can the synthesis of CDKs and cyclins, we can monitor that. And finally, the action of specific, there's CDK inhibiting proteins out there. So the use of those could regulate the cell cycle as well. But the activation or inactivation of these CDK molecules is what produces the signals pushing the cell through the cell cycle. And again, we want to do this in a specific, regulated manner to avoid catastrophe such as cancer. So if we look at the first method, regulation of CDKs by phosphorylation. The residues that are mainly affected here are the threonine 160 and tyrosine 15 residues of CDK2. But basically when threonine 160 is phosphorylated, its conformation is going to change a little bit, and it opens the catalytic site on the CDK, allowing for, it does that by moving an auto-inhibiting T-loop away from that binding site. The other one, the tyrosine 15, when tyrosine 15 is dephosphorylated, it removes a negative charge that's blocking ATP molecules from reaching the binding site. So the ATP is able to get there, they can activate the CDK molecule. So tyrosine 16 kind of is an inactive state when it's phosphorylated, whereas threonine 160 is activated when it's phosphorylated. And we can see that in the diagram here. So we have cyclins produced, and the cyclin CDD forms, but phosphorylation on tyrosine blocks the ATP binding site, so it's still inactive. Once we get the phosphorylation of threonine 160 and the T-loop removal, then that tyrosine 16 can be dephosphorylated, and we kind of move through the process and continue there. Removing of the negative charge and allowing the ATP to bind to the tyrosine 16 or 15 allows for activation of molecules downstream from this. So phosphorylation of things like the PTP molecule. We use that to phosphorylate downstream protein targets in the cell cycle. If DNA damage is detected, the cell cycle can be stopped in this position in G2 by the activation of two proteins, ATM and ATR. These two proteins cause the inactivation of PTP, which is what dephosphorylates the tyrosine 15 of the CDK. So the inactivation of CDK is what stops the cell cycle in the G2 phase until that DNA damage can be repaired. The next way we can regulate the activity of the CDKs is through the controlled degradation of cyclin. Since the CDKs are essentially only active when cyclin molecules are attached to them, the degradation of cyclin molecules can serve to regulate the cell cycle. They must be degraded at specific points, since particular cyclin molecules activate specific steps in the cell cycle pathway. So cyclin molecules contain a destruction box of a particular amino acid sequence near their amino terminus end of the protein. Now, the destruction box recognizing protein, or DBRP, recognizes this destruction box sequence, and that initiates, when they combine there, that initiates the degradation of the cyclin molecule by linking the cyclin with a ubiquitin molecule. Okay, we're kind of seeing that in the picture here with step 7. I know it's a little blurry, and I apologize for that, but we can see when we activate the DBRP with phosphorylation from the CDK, it activates it, and that triggers the combination with ubiquitin, and then we get what we see here. So linking with cyclin with ubiquitin molecule, that complex is joined together, and we can add multiple ubiquitin there, and we end up with a proteasome, and that proteasome can degrade the cyclin. So we see that here in step 8. The rate of the cyclin breakdown is controlled by a feedback loop, and that's what we see here in this region. As CDK activity increases, cyclin proteolysis is initiated. The production of cyclin and attachment of a CDK molecule causes the phosphorylation and activation of DBRP. This feedback loop ensures that the cyclin molecule and CDK are present and activated when they are needed, and they are inactivated when they are no longer needed. Okay. The next method we can use to regulate our CDKs is through growth factors stimulating CDK and cyclin synthesis. So different CDK molecules have different, sorry, I apologize there. The cell cycle can be regulated by manipulating the rate at which both CDK and cyclin are produced. So if we need these factors to make the cell cycle go, as we produce them, we're going to control whether the cell cycle is going or not. So growth factors and cytokines both can use phosphorylation to activate various molecules. In this case, they're going to phosphorylation the Jun and Phos nuclear transcription factors in the nucleus. Activation of those through phosphorylation then induces the production of E2F, and the production of E2F triggers the creation of nucleotides and DNA. The cyclin and CDKs that are triggered by this transcription factor allow the cell to enter the S phase so that they can use the DNA and enzymes produced for DNA synthesis here to produce copies of DNA. Okay, sorry about that. Okay, the last way we regulate it is through inhibition of CDKs. Um, Different CDK molecules have different inhibitors that can bind to them and inactivate them at various parts in the cell cycle. According to McHale, Albanese, and Pishovan in 2015, CDK4 and 6 inhibitors include the molecules PD033291, LY2835219, and LEE011. I know those names don't mean a whole lot to a lot of people, but we can see that here. So it inhibits these, which affects this part of the cell cycle. We can see other ones here like P15, 16, 18, 19, P21. So there's various inhibitors that will affect CDKs at various points in the cell cycle. Uh, by combining with the CDK molecules, in some ways, the inhibitors prevent the phosphorylation or dephosphorylation of the CDK molecules. So inactivating these molecules affects the progress of the cell through the cell cycle. These inhibitors are being studied for their use as potential anti-cancer treatments, which will be discussed later in the presentation. Basically, in cancer cells, sometimes these CDKs get stuck in the on position, pushing the cell cycle to move forward. So utilization of these inhibitors has some promise in deciphering or trying to prevent cancer from happening. All right. There are several proteins or critical proteins that CDKs work with within the body, and we're going to look at a couple of those now. The first one is lamin. Okay, if we look at this one, lamin is a protein that is one of the main components of the meshwork of the filaments that go together to build the nuclear envelope. So if we look in the drawing here, we can see that M-cyclin CDK in prophase, we want to break down that nuclear envelope. And then again, when we get to telophase, we want to start building that back up again. So lamin is actually depolarized when it is phosphorylated by a CDK molecule, according to Nelson and Cox. So we see that here. The M-cyclin CDK phosphorylates it, and it's able to break down the nuclear envelope. This contributes to that breakdown of the nuclear envelope during mitosis and allows the sister chromatids to spread to other sides of the cell, and then we reform those nucleuses when we're done. So we dephosphorylate the lamins between prophase and telophase so that once we get to telophase, we can start building those nuclear envelopes up again so that we have nuclei surrounding both cells. Okay. The next compound that we see CDKs affect is actin-myosin. Mostly the myosin is what they're going to target here, but we want to talk about both of these together here. So what we see in the picture here is cytokinesis occurring. During cytokinesis, you see an actin-myosin ring here around the center. And actin and myosin is the same contractile mechanism you find in your muscle cells. And what happens here is they slide past each other, sliding filament theory, lock into place, and the contraction of it causes the cleavage furrow to form and eventually pinches the two cells into two separate cells. This process is driven by the actin-myosin machinery requiring the use of ATP. Now, CDK is responsible here for inactivating this contractile mechanism. So it does this by phosphorylating a subunit of myosin, which causes the myosin to disassociate with the actin filament. By doing that, it can reset, and when the cell cycle comes up again to this point where cytokinesis needs to occur, the mechanism is in place for it to do that again. The next protein discussed is the retinoblastoma protein, or PRB. In this case, obviously named retinoblastoma, it was discovered first in retinoblastoma tumors. And if we look, what it's going to do, it's going to halt the cell cycle in the G1 phase. It does this when it notices DNA damage. So if we look in proliferation, okay, in senescence, we can see it activates some different things there. depending on what's going on. This protein was first discovered in, like I said, the retinal tumor cells, but it functions in virtually all cells. When it is unphosphorylated, it will bind to the E2F transcription factor, and that prevents DNA synthesis. Okay, so when it's unphosphorylated, it bonds to the E2F, and DNA synthesis does not occur. We have that pathway blocked there. However, when it is phosphorylated, the cyclin E CDK2 can phosphorylate the PRB, allow it to detach from the E2F transcription factor. And when that occurs, that allows the cell cycle to continue. So we can see the first where it's inactivated because it's unphosphorylated. The E2F and the retinoblastoma protein are combined, and the cell cycle signal is not being sent. However, we phosphorylate the retinoblastoma protein, it detaches from the E2F, and it produces or allows the E2F to then go and transcribe the synthesis of the DNA nucleotides and stuff that it needs to cause DNA to synthesize. So we would get stuck here in the G1 phase until we phosphorylate the retinoblastoma protein, and then it allows us to move into the synthesis phase. This is important because if DNA damage is noticed, this mechanism here gives the cell time to go through and repair the DNA before it continues with the cell cycle. Okay. P53 CDKs in cancer. One of the last thing I wanted to talk about was how some of these CDK molecules can be used in cancer treatments. Now, P53 is probably the most well-studied and common tumor suppressor gene located in human cancer patients. So it's found in many cancers. When functioning properly, P53 serves as a transcription factor and causes the cell to produce P21. So we kind of see that here. And the production of P21, according to Shaw, inhibits multiple cyclin-dependent kinases. So this can go through and it has effects on multiple CDKs here. The CDKs most affected are CDKs 2, 3, 4, and 6. So inhibiting these stops the cell cycle in the G1 phase. So that's how the P53 serves as a tumor suppressor gene, producing the P21, which inactivates the CDKs, pausing the cell cycle in the G1 phase. A second means of P53 regulation comes from the ability to regulate the expression of cyclin A. By regulating cyclin A, the CDKs need that cyclin in order to have an active space for things to go. So therefore, damage here causes the cell cycle to slow down as well. Now, since P53, if we look in the picture here, since it has such a big step on the cell cycle, and even the retinoblastoma protein, E2F, those are all downstream from where this enzyme works, or this protein works. So by affecting that, damage to the P53 gene can affect the regulation of the cell cycle by failing to inhibit those CDKs and producing cyclin A. That would result in the continuation of the cell cycle under conditions where it would normally be stopped. So an unregulated cell cycle can result in cancer. So the objectives were to explain the phases of the cell cycle, to describe why the cell cycle is regulated, describe the structure and function of the CDKs, to explain the 4 methods of CDK activity is regulated, to provide examples of how CDKs affect critical proteins, and how CDKs are linked or used in cancer treatments. I think we've accomplished most of those, and here are my references, and I thank you for your time.